that I'd already gone over. And I'm going to go into a second research. This is slightly indirect, and it's a lot of its results are assumed, okay, but it is relevant to genetics and behavior in terms of epigenetics. If you can remember what we studied in genetics and behavior, epigenetics is what is beyond the gene. Okay, it's the environment. How does the environment um, either help activate a gene or suppress a gene? Okay, so that's epigenetic influence. Okay, so Meany et al., I think that's how you say it, in 1988, again, a pretty old study, aimed to see the effect of stress hormones on memory by studying newborn rats. Okay, it was an independent measures design with two distinct conditions. In condition one, uh, researchers separated newborns from their mothers and placed them in a plastic container lined with a paper towel, which was a stressful situation. So, being separated from the mother and being put in such a confined area is a stressful situation because they're not able to escape. Okay, but in this condition, when the rat was in the container, they were brushed for an intense 15 minutes by the um, researcher, which basically simulates the same grooming that a rat's mother would do. Okay. On the other hand, in condition two, they were also taken away for 15 minutes from their mothers and put in the container. But these rats received no stroking. Okay, so the researchers did not stroke these rats, and they just left these rats in distress in that container for 15 minutes. Okay, which would have induced high levels of anxiety or stress, rather. Okay. Uh, so this procedure was conducted daily from birth till the mice or the rats were in a position that they were weaning away from their mom's breast milk. Okay, so this was the duration of which they conducted this procedure. So to test the effect, okay, of the elevated stress over their lifetime, at the age of 2, the rats were put in a pool of milky water, which had a platform to escape. Okay, so till now they must have put these rats in water before to just see what their reaction would be, and now they have at the age of 2 to check whether the effects of stress was long term. They have put them in a glass of or container of milky water with a platform where they can escape through. So once they were put in that platform or in that milky water, the researchers observed and tracked the route that the rats would take to escape the milky water and get onto the platform. Okay, what they found was that the high levels of stress hormones in early life resulted in neuron loss and cognitive impairments in the rats at an older age. Okay, the rats that were neglected, basically not stroked when they were put in that container, took longer routes to get to the platform. So this was the route that the um, rats that received the grooming and stroking took. It's pretty straightforward, and this is the route that the ones that were not groomed took to escape the water. Okay, so it shows that the high levels of stress in early life impaired their cognitive ability and also caused cell death in their hippocampus, if I'm not wrong. Okay. So these deficits, these cognitive and neuronal deficits, were not absent. Were absent in the groomed rats. So the ones that were groomed, that were given the care, they did not see these cognitive and neuronal deficits in them. Okay. This research then suggests that extra grooming leads to the activation of genes responsible for one's reaction to stress. Okay. For example, it is also a very good example of epigenetics, where grooming is the external environment that has turned on genes which help the rat cope with stress. Okay, we can see then the interaction between environment and genes is very important in manifesting certain behaviors. Okay, so long-term exposure to cortisol, which is the stress hormone, leads to overstimulation, okay, causing hippocampal cell death. So when there's hippocampal cell death, it affects the ability that we have and that other animals have to create memory, okay? Because the hippocampus has acetylcholine, which is a neurotransmitter responsible for memory as well, um, and when there's cell death, there is lesser ACH in the hippocampus. It results in cognitive impairment in terms of memory. So because this happened, there was cell death. There was cell death, and there was cognitive impairment as a result of that. Okay, and there's ACH involved, and there's stress involved. Okay, in this group that was not groomed. So when there's lower ACH and there's higher stress hormones, they have found also correlations or individuals with Alzheimer's to have similar trends in these chemicals in their brain. Okay, so this supports Meany et al.'s research on epigenetics. So if you have higher stress in your life, there can be atrophication of your hippocampus, which can eventually lead to Alzheimer's. Okay, similar findings have been associated with kids who have been abused in their childhood. Basically, hippocampal atrophy is seen in children with abuse because they have experienced stress really early on in life, and same atrophication or a reduction in volume was seen in veterans with PTSD who again have experienced really high levels of stress. Okay, in terms of evaluation, it's a lab experiment. Again, it allows us to develop a cause-effect relationship between the grooming and the switching on of genes and how that relates to cognitive ability and coping with stress. Okay, but the question is, does this procedure translate into real life? Okay, and they have found, like the examples that I gave, ways in which this is relevant to human beings and even real life. And in terms of ethics, the rats were killed here as well to check the levels of neurotransmitters that they had, which is unethical. But if it is serving a utilitarian approach or purpose, which means that it helps the greater good in understanding epigenetics and polymorphism, okay, and it helps us understand human behavior and animal behavior, then it is, in terms of a cost-benefit analysis, it is beneficial to science and research and psychology. Okay, so with that, that's my lecture for today. Again, like I said, this is genetics and behavior. You will be looking at brain and behavior and hormones and behavior. Again, you will go with aim, procedure, the participants obviously being which animal they used, the results that they found, the conclusions, how that relates to human behavior. It is very important for you to relate it to human behavior. So if you're looking at Rogers and Kestner's ACH study, you know that there's Antonova's research on ACH as well. So you need to find relevant research that is associated to these animal models, because if you just talk about the animal model and do not associated or related back to human behavior, you're not going to be able to get the top marks. You probably will get really low marks, in fact. Okay, so pick 2 researches per topic, write it down as an evidence bank for now, and do the evaluation in terms of the procedure, but also in terms of the ethics, and how that relates to the greater question of whether animal models are good to use for understanding human behavior or not. Okay, same for hormones and behavior. That will be your task for today. We will continue to work on this on Monday and start doing essays. I want our class to really have essays for this topic and other extensions down, so you are well prepared to answer questions on the extensions that we have for each unit. Okay, bye-bye.